Hello everyone, I'm Anju Sunny. First of all, I would like to thank the ECR committee for providing me this opportunity to present my research. Uh, I'm a PhD candidate at University of Alabama in Dr. Wu's lab, and I will be graduating this August. I have done my master's from Mahatma Gandhi University, where my thesis focused on natural product isolation and small molecule synthesis. After my master's, I have joined for the PhD program in University of Alabama, where at Dr. Wu's lab, my research focuses on the development of novel top-down methods for proteoform functional characterization, as well as the application of label-free quantitative top-down proteomics methods, especially to the aging studies. Through this work, I have developed a strong interest in structural proteomics, particularly in studying aging and neurodegenerative diseases. I am currently pursuing postdoctoral opportunities in this field, and my long-term goal is to build a career in the analytical R&D in pharmaceutical industries. So today I will be presenting our recent paper, Methionine Oxidation Footprinting in Intact Proteins, abbreviated as MOFIP, using top-down proteomics. As we know, the protein are dynamic molecules whose structure and functions are influenced by solvent accessibility, stability, as well as interactions with other biomolecules. And proteins has like 4 levels of structural organization. The biological activity of a protein is intrinsically linked to its tertiary as well as the quaternary structural organization. Classically, there have been multiple methods that has been used to study the protein structure, and most of them give high resolution structures. But however, these methods are limited on giving information regarding PTMs as well as providing the endogenous information regarding the endogenous protein structure. More recently, mass spectrometry methods has been used to understand the protein structure and function. Among the mass spec methods, protein footprinting methods are typically used for studying the protein structure and its function. The two most well-known protein footprinting methods are hydrogen deuterium exchange mass spectrometry as well as the hydroxyl radical footprinting mass spectrometry. In hydrogen deuterium exchange, proteins are treated with D2O and the amide hydrogens exchange with deuterium based on the condition. Similarly, in hydroxyl radical footprinting mass spectrometry, the proteins are treated with hydroxyl radical and the amino acids which are solvent accessible reacts with the hydroxyl radical. But typically, these methods are applied to pure proteins or simple protein mixtures and commonly employed with bottom-up proteomics methods. The solvent accessibility of a protein is very important to study because they have significant role in the protein structure and folding, and they give information regarding protein-protein interactions as well as the active and binding site of the protein, also the allosteric regulation. Thereby, the solvent accessibility connects the dot between a protein's 3D structure as well as its biochemical behavior. As we have seen, the hydroxyl radical footprinting methods focus on like a lot of amino acids because the side chains of many amino acids can react with the hydroxyl radical. But whereas a method that can probe the methionine oxidation at the proteoform level will allow the examination of the role of the methionine residues in protein structure, in protein stability, as well as its function. The key reaction here is methionine is allowed to react with hydrogen peroxide. So it will form the methionine sulfoxide. By making use of this reaction, we can extend this method to study the solvent accessibility of methionine residues, as well as if we couple the hydrogen peroxide reaction with denaturation, we can study the buried methionine residues as well. So here we have developed like a novel top-down method that help us study the solvent accessibility of the methionine residues in proteoforms. Here we call it methionine oxidation footprinting in intact proteins. So for the proof of concept, we have used E. coli cell lysate. So the E. coli cell lysate is treated with and without hydrogen peroxide. After a fixed oxidation time, the oxidation reaction is quenched with the addition of L-methionine, followed by a 10 kilodalton molecular weight cutoff filtration and buffer exchange to get rid of all the MS incompatible salts, followed by the LCMS analysis. Let's consider like an ideal scenario where a protein is having one methionine residue. It can have three possible outcomes. Upon reaction with hydrogen peroxide, if it's located outside as shown in the structure, it is solvent accessible, so it can undergo reaction with hydrogen peroxide, so it will get oxidized. So as you can see from the results, in the sample without hydrogen peroxide, only the unoxidized species will be there. In the sample with hydrogen peroxide, only the oxidized species will be there. The second case, where a protein is having a methionine residue and is located inside, since it's buried, it cannot undergo reaction with hydrogen peroxide, so the sample with and without hydrogen peroxide will only have the unoxidized form. Whereas a third scenario is possible where a protein can have a methionine residue and it can exist in free as well as bound population. So upon reaction with hydrogen peroxide, only the protein which is free can undergo reaction with hydrogen peroxide, whereas the bound one remain as it is. So in the hydrogen peroxide sample, we see both the oxidized as well as the unoxidized form. Coming to the real results, autonomous glycyl radical cofactor protein is an E. coli protein. So as you can see from the structure, it has only one methionine, which is methionine 1, the N-terminal methionine. Upon reaction with hydrogen peroxide, we completely see the oxidized form, and this is the relative abundance plot from the replicate sample is also shown here. As you can see from the bar plots, it's consistent. And another example of a solvent inaccessible helix is a phosphocarrier protein. That's another E. coli protein having two methionine residues, methionine 1 and methionine 81. So methionine 1 is the N-terminal methionine, whereas the methionine 81 is located in a helix. Upon reaction with hydrogen peroxide, we only see a single oxidized species as demonstrated in the bar plots as well. Further, from the MS2s, you can see the single oxidation is localized to the N-terminal methionine. So upon hydrogen peroxide treatment, only methionine 1 is getting oxidized. The methionine 181, which is located in the helix, is buried and its solvent inaccessible. Cold shock protein is another E. coli protein. So it has only one methionine residue, methionine 5, which is located in a sheet. So in the sample with hydrogen peroxide, we see both the unoxidized as well as the singly oxidized species. Furthermore, we have done another experiment where we have treated with the sample with 6 molar guanidinium chloride followed by hydrogen peroxide oxidation. But we see similar results even in 6 molar guanidinium chloride with hydrogen peroxide. We see the unoxidized as well as the oxidized species, suggesting this methionine 5 is partially accessible. And this cold shock protein is an RNA binding protein. So in the native E. coli cell lysate, it is a possibility that both free cold shock protein and the RNA bound protein is coexisting. Overall, we could quantify the solvent accessibility of almost 69 proteoforms, and among the profiled methionine residues, the 45% were in helices, 20% were in sheets, and 35% were in coils. So, among the methionine residues in helices, 51% were solvent accessible, and among the methionine residue in sheets, 29% were solvent accessible, and 63% of the methionine residues on coil were solvent accessible. So, to summarize, our MOFIP method enables the determination of solvent accessibility of individual methionine residues in intact proteoforms from complex biological samples. Also, we could observe the solvent accessibility changes that arises from the structural changes in proteoforms, which is often challenging to study with bottom-up approaches. And our results suggest the partial accessible methionine residues in intact proteoforms, which can be further investigated to study the solvent accessibility of the proteoforms in the native cell lysate. Thank you.